Welcome back to Binary Bio, where we bridge the gap between computer science and the building blocks of life. What if I told you that the same algorithm used to decode the human genome was invented in 1970 and fits in a single grid? That's Needleman-Wunsch, and today we're going to build it from scratch. Needleman-Wunsch solves one problem. Given two sequences, find the best possible way to line them up. Not just partially, globally. Every single character from start to finish gets placed. Matched, mismatched, or gapped. In biology, this is how scientists compare DNA, RNA, and protein sequences. Want to know how similar two species genes are? Trying to spot a mutation? This algorithm is your starting point. It's been foundational to biological research for over 50 years. Here are our two sequences, GCAT-GCU and GATT-ACA. Both are 7 characters, so our matrix will be 7 by 7. That's 49 cells to fill. Before we start, we need a scoring system. A match scores +1, a mismatch scores 0, a gap also scores 0. Simple, 0 penalties. Let's build the matrix. For every cell, we look at 3 neighbors and take the max. The diagonal gives us a match or mismatch. Both sequences advance together. Left means we insert a gap in sequence 2. Up means we insert a gap in sequence 1. Let's fill the first row. G versus G, match. Diagonal equals 0 plus 1, so the score is 1. G versus C, mismatch. Diagonal is 0, but left carries a 1. Score stays 1. The rest of the row fills the same way. Every cell picks up the best available value from its neighbors. Each row builds on the last. Matches push the score higher. Gaps carry the best score forward without losing ground. After all 49 cells, we reach the bottom right corner, and that value is our optimal alignment score. The best possible global alignment of these two sequences earns 4 match points. But the score alone doesn't tell us how to align them. That's what the traceback does. Starting from the bottom right, we follow the arrows back to the origin. A diagonal move means both sequences advance, match or mismatch. A horizontal move means a gap was inserted in one sequence. A vertical move means a gap in the other. Follow the highlighted path and you get the final alignment. Two sequences perfectly placed side by side. That's Needleman-Wunsch, a 54-year-old algorithm built on a grid that still powers modern bioinformatics today. Now that we know how Needleman-Wunsch algorithm works, let's solve a Rosalind problem. In this problem, we are given two accession IDs, JX46999.1 and JX308819.1. These are identifiers for DNA sequences stored in the NCBI database. Our goal is to fetch these sequences and compute their global alignment score using the Needleman-Wunsch algorithm. So the problem has two main steps. First, we need to retrieve the sequences from NCBI. Second, we need to align them globally and compute the score. Let's walk through how the solution works. We start by using Biopython, which is a powerful library for working with biological data. It gives us tools to fetch sequences from online databases and perform alignments without writing everything from scratch. The first thing we do is set an email address. This is required by NCBI whenever we send a request to their servers. It's just a way to identify who is making the request. Next, we define a function whose job is to fetch a sequence given its accession ID. Inside this function, we send a request to NCBI's nucleotide database and ask for the result in FASTA format. FASTA is a standard format for representing sequences. Once we get the response, we read it using Biopython's sequence reader. This gives us a structured object that contains the sequence. From that object, we extract just the DNA sequence and return it as a simple string. So now we have a helper function that takes an accession ID and gives us the corresponding DNA sequence. Next, we define another function to compute the alignment score between two sequences. Inside this function, we create something called a pairwise aligner. This is the tool that will perform the alignment. We explicitly set the alignment mode to global. This is important because the Needleman-Wunsch algorithm performs global alignment, meaning it aligns both sequences from start to end. Then we load a substitution matrix called NUC.4.4. This matrix defines how we score matches and mismatches between nucleotides. Instead of manually assigning scores like +1 or -1, we use a biologically meaningful scoring system. After that, we define gap penalties. Opening a gap has a higher penalty, and extending a gap has a smaller penalty. This reflects the idea that starting a gap is more costly than continuing one. We also set penalties for gaps at the ends of the sequences. By keeping these consistent with the internal gap penalties, we ensure that the alignment remains fully global. Once all the scoring parameters are set, we compute the alignment score between the two sequences. The aligner automatically finds the optimal alignment using the Needleman-Wunsch dynamic programming approach we learned earlier. It considers all possibilities and returns the best possible score. Finally, in the main part of the program, we define the two accession IDs. We fetch both sequences using our helper function. Then we pass those sequences into the alignment function and print the final score. So overall, this solution combines two ideas: fetching real biological data from NCBI and applying the Needleman-Wunsch algorithm using Biopython. What's nice here is that we don't have to manually build the dynamic programming matrix. We already understand how it works conceptually, and Biopython handles the implementation for us. Behind the scenes, it's still doing exactly what we learned: comparing characters, applying match and mismatch scores, adding gap penalties, and building up the optimal alignment step by step. If you found this breakdown helpful, make sure to hit that subscribe button and ring the bell. Thanks for watching, stay curious, and keep decoding. I'll see you in the next one.